Okay, it's 10 o'clock here in Oregon, so we're going to get started. I'm your host, Alice Formiga of eOrganic, and I'm very pleased to be able to host this webinar on breeding carrots for production, resilience, flavor, and fun in organic systems. And before we begin, I just wanted to let you know that we're recording this webinar and it will be available along with some other resources and a handout of the slides on the eOrganic website at eOrganic.org. And also the recording will be available on the eOrganic YouTube channel. So I'm very pleased to welcome our team of carrot breeders. We have Solveig Hanson of the University of British Columbia, Phil Simon of the USDA ARS in Wisconsin, Laurie McKenzie and Micaela Colley of the Organic Seed Alliance. So Laurie, I'm going to give you the remote control here.
All right. Good morning, everyone. Thank you so much for joining us. We're all really excited to be here with you today and share our carrot knowledge and experience and hear some of what you all have going on. So I'm just going to start us off with sort of a laying foundation of what we're going to talk about, and then some terminology, just so we've all got common language. It's probably not going to be super new information. And then we'll get into some of the really fun stuff about selection and color genetics and trait genetics. I'm going to present first, and then Solveig will talk about selection, and then Phil will talk about color genetics and traits. We'll all kind of pop in on each other if we have things to add. So it'll be a little more of a conversation, hopefully, than just a straight up presentation. So this is breeding in a nutshell. Really, it's finding things that you like or that you're looking for and selecting them and grouping them together, allowing them to cross, and just doing that over and over again, essentially. So imagining that these are very well-known terms, open pollinated and hybrid, we're not going to really focus on either of these today, but as OSA, the OSA Research and Development Breeding Education team, we focus most of our energy on open pollinated carrot breeding. We're dabbling a little bit with hybrid breeding, and we may do another webinar to get into that. This is a really foundational and key concept for today, that the phenotype, what the plant looks like, what the roots look like, is a combination of the genotype, which is the genetic makeup of the plant, interacting with the environment. And that's always true. So both the environment and the production system and have significant impacts on the phenotype. And we see this when we go down to El Centro in Southern California with Phil to dig and evaluate the winter nursery. The roots there don't always look the same as they do when we grow the same lines and the same populations up here in Northwest Washington. So keep that in mind in your own breeding work and as we go through and talk about our experiences today. And not everything that you see will be passed on to the progeny or the next generation. So that can be really frustrating when you find something that's really exciting and beautiful. You get a good seed crop off of it, and then the next year you don't see it anymore. Hopefully we'll address that a little bit as we go through the concepts and experiences today. Getting a little deeper, genes. Genes are a unit of inheritance that controls a given trait, and genes exist at a fixed location, or also known as a locus or a loci on a chromosome. And an allele is a possible variation of a gene. And depending on the gene itself, sometimes there's a lot of alleles, sometimes there's tens or hundreds, sometimes there's a handful. We won't get too much into F1, F2, F3, but that's an abbreviation for filial, which is just indicating the generation after a cross. So F1 is the first generation after you cross something, F2 is the second generation, F3 is the third. Heterozygous means having two alleles that are non-identical or different at a locus. Homozygous means the alleles are the same. If you have a dominant allele, then that will produce the given phenotype regardless of whether you have one copy or two copies of that gene. If it's a recessive allele, that means you have to have 2 copies for the expression to be seen. So we're going to talk about trait inheritance as also kind of a foundational concept today. And it's on a spectrum and a scale from kind of simple to complex. We're also taking concepts today that are big, dense concepts and trying to kind of tackle them in a way that's accessible and maybe a little more straightforward than they really are. So just kind of keep that in mind. It's like life. Everything is complex and has lots of factors in play. So when we talk about sort of simple trait inheritance or simple traits, we're talking about things that are controlled by one gene or maybe a couple of genes. And these, when you see these phenotypes in the field, they usually are distinct. So think of Mendelian genetics of like the white flower, the red flower, and then the pink flower when the red and the white cross. That's what we mean by distinctive categories. And usually expression of simple traits is not as environmentally sensitive or variable. So if you've got 2 genes for an orange carrot, you're going to get an orange carrot. It may vary a little bit from location to location or environmental condition, but it's still always going to be orange. And then when we get to the other end of that, and we're talking about more complex traits, these are traits where generally many genes are involved in the expression. And these are traits like flavor, disease resistance, salinity tolerance, abiotic and biotic stress tolerance. So things where there are a lot of genes at play, a lot of genes interacting for those responses. And in this, instead of seeing sort of distinct categories, we see a range or a spectrum of phenotypes. So instead of just white, pink, and red, we're going to see white to light pink to medium pink to dark pink to red. And that expression tends to be a little more environmentally responsive and sensitive depending on what's happening in the environment. And with that, that's kind of the brief and quick foundation. I'm going to hand it off to Solveig to talk about kind of how to do selection and some techniques and tips when you are thinking about what kind of selection and how to do it.
Yeah. Hi, everybody. Nice to talk to you. Before we talk about selection, you just have to say that we're selecting from a variable population or a population with a lot of different trait expressions for the traits you're interested in. So if you're interested in the flower color that Laurie was talking about, you want to make sure you have lots of different flower colors to select from. So that's kind of a prerequisite. But once you have this population, and also start talking about carrots, since this is a carrot breeding webinar, if you have a big variable population that you've created somehow by crossing maybe a couple of parents or populations that have different traits that you want in your final variety or population, first thing you're going to do is select vigorous individuals. So you want like stecklings or roots, like we're selecting there on the table, that will survive. So healthy roots. You want to consider both that the traits you see are results of both genetics and field conditions. So yes, you want to select individuals that look great, but also think about how the environment, your field, might affect the traits that are showing up. Think about disease and pest pressure. To select for resistance to a disease or pest, you have to have pressure from that disease or pest. And if you don't, if you haven't had that season for whatever reason, that's not going to be a great year to select for that.
Yeah.
Go ahead.
Jump in for just a second. So one thing to consider if you do want to, you have a disease on your field, let's say you have powdery mildew, and you have a variety that you know is susceptible, and you are working with some breeding material that you're not sure is, you can plant the susceptible variety in with it so that you can see that you have the disease present, and you also have essentially an inoculant right next to your plant, so you know they're getting exposed to it. So you can kind of manufacture the pressure for disease if that's a high priority for your breeding work.
And if I can enter one point here, please. Solveig, you mentioned the term steckling, and that refers to the top. These are stecklings laying on the table with the upper portion of the carrot root, along with somewhere between a centimeter and an inch of the petioles, and that's what's used to store and make the seed crop the next generation.
Perfect. Yeah, thank you both. Those are good clarifications. And then once you move out of the field, so you've selected the individuals that look vigorous and healthy, then it's time to think about an ideotype, or what shape are you looking for in your carrot, since shape determines a lot of carrot market class. Like, do you want a nonce type that has kind of cigar shape and has a blunt tip? Do you want a more tapered type? Do you want a ball type carrot? Things like that. Selecting for smoothness, lack of root hairs, things like that. And then finally, it's time to taste, and we'll talk a lot about tasting. And I think something Laurie emphasizes a lot, and Phil, is to taste all throughout your selection process. Like, instead of saving it till the very end before you're ready to try to go to market with something that looks perfect, it's good to taste along the way to make sure you have great flavor traveling along with the beautiful color. Okay, so there are a couple of ways we're going to talk about to approach selection. And the first is mass selection. It's the simplest. It means you keep track of one population, you plant it out, and you select the individuals that look the best on all those criteria we just talked about. It's important to have as large a population as you can handle, that you can grow well, so that you have a lot of individuals to select from and still end up with enough individuals in your population to avoid inbreeding depression when you intermate them. It's important to choose a site with as uniform soil conditions as possible. If you plant out your population and realize that you don't have a very uniform field, you can also divide it into sections or quadrants, and try to select evenly the best individuals from each quadrant to mitigate those environmental effects. And the drawback to mass selection is that each individual doesn't give you any information about what genetics might be hiding in it, what recessive traits it might be harboring that will come out in future generations. So you don't make progress as quickly toward the traits you want if you don't take into account what an individual's relatives have to say. But it is a simple way to approach selection, and you sure can make progress. Another way to do selection is family selection. This is from our Canovi carrot plot up at University of British Columbia this season. And so to do this, we saved seed off each individual plant that was in our seed population. We planted them all out in separate rows, harvested them all separately, and then first selected families that looked good. So we took all the information from all the genes that were, or the phenotypes that were present in a certain family, and said, we like these 15 families out of the original 50 the best. And then we can go on to select within that family for the best individuals. So it's more time consuming, takes some record keeping, but you can make progress faster toward your goal. These are some photos from our process. So this is how we phenotyped each family in terms of marketable, unmarketable, and we were really interested in carrot rust fly, any carrots that had lesions. And then from within this family, these are the individuals we decided to take for our final population. Little summary, mass selection is slower, but it's simpler. You keep track of one population rather than as families, but you have less information with each individual because you don't know what recessive genes might be hidden in there that will show up in future generations. So to make progress with selection, you need variation from for your trait of interest in your original population. Heritability, or the amount of the trait that gets passed to the next generation, will vary with the trait. So that's a great thing to observe and do research on, or ask one of us who has more experience with carrots, and keep in mind that your selection strategy will determine how much information an individual gives you about any other individuals it's related to. When you think about how much to select, it's basically a game of balancing how much progress you want to make, how fast, with creating a big enough population to avoid inbreeding depression.
And I would say how expressive your trait is. So if you have a trait where, let's say you have 100 carrot roots and the trait only shows up in 5 roots, then that's this 5% selection intensity. But moving forward with 5 roots is kind of a genetic bottleneck, depending on how much diversity is already in the background. So if you have a trait that you like and is only shows up in small amounts, that's when you need to plant a big population. So maybe you plant 1,000 roots, you get 50 that have the trait that you want, you take those and you move those forward. So this is just sort of a visual representation of kind of you're always going to have a bell curve when you're doing mass selection. And, really anything kind of, nature is dynamic. The bell curve might be really, really smushed up if you get really genetically uniform. But what we're really trying to show you here is when you make selections out of a population, you move it in one direction. And the harder you select for something, the bigger the jump can be.
So basically, since traits are controlled by both genetics and environment, even if you select this 5% of phenotypes that you love, because some of that expression is environmentally controlled, the mean of that trait in the next generation is going to be somewhere between the mean of the original population and the mean of the individual you selected.
Yeah.
So just to gauge your expectations, and now we'll go back to Laurie to talk about some cool color populations. Oops.
Yeah, so I'm just going to run you through how this actually works in practice, or it has worked for us. This is a current project that we're working on. It's one that we started in 2018. And when I first went to the winter nursery with Phil back in 2015, 14-ish, my mind was just blown wide open by the color and the diversity and just the astonishing amazingness that I saw in these 1,700 plots in his winter nursery. And I just was absolutely captivated, especially by the purples. And I just couldn't stop myself, honestly, from bringing them home. And so up here in the upper right, you see this trio. This was my favorite phenotype. And every time I cut carrots open, I think I took 600 pictures, and probably at least half of them were that same phenotype. And so I collected some roots of several different plots of Phil's breeding materials, and I brought them home. Brought them home, put them in a cage all together. This is the very first generation in 2018 from that cross. And then in 2019, I added in some roots that look like this because they were incredibly crisp and delicious and beautiful. They weren't quite as purple, but they also had this just really dark purple center, which continues to be my favorite to this day. And you can see it changed. So we went from, fairly uniformly dark population, and then we started seeing a lot more yellow showing up in 2019. So this is what that looked like. And we laid it all out and we thought, well, this is interesting. We've got these nice dark roots. We've got these kind of more raspberry roots. And now we're starting to see these yellow roots with some kind of purple bruising. And maybe it would be fun to just have this variety that's diverse, that we work on the shape uniformity, we work on the length uniformity, but we let the colors kind of stay diverse. We thought about that. And then we thought, well, no, let's split it because we can always kind of mix it back together if we want to. So from that 2019 population here on the top, we split it into a lighter population and a darker population. So in 2020, this is what we had. So in 2019, then we had 2 separate pollination cages, one with these darker phenotypes and one with these lighter phenotypes. And in 2020, we did that again in 2020. And so in 2021, our PYP light population is starting to just overall look a lot lighter. We're getting a lot more of this yellow expression. We're starting to see a little bit of orange pop up. So we've got these much lighter kind of raspberry purple, a lot of yellow, still looking for that dark purple core. We're parceling off these orange ones into another third population. Here's where we're at with the PYP dark. This was Micaela, Phil, and I at the winter nursery just a couple weeks ago, actually, going through this population. So you can see here on the table, this is much darker. We've still got some yellows, as we'll learn from Phil here in a minute. Yellow and purple are pretty good friends genetically, so it's hard to separate all of that yellow out. But we're at a point now where we are probably with both the dark and the light population. I'm going to put these all in a cage this year, and then I'm going to do family selection next year. So we'll save seed from each of these roots individually. We'll send them down to the El Centro Winter Nursery in October, and there'll be a plot from each one of these roots. And then we'll look at what we get out of each of those plots. Do we get something that looks like this, that still has a fair bit of color diversity, or are we starting to get something that looks a little more uniform? Hopefully we'll start seeing plots that are giving us something that's tending towards this in more of like the 80% range. And we'll take those plots and we'll take roots from those plots and we'll continue forward with those in the next generation. So we'll do that with both these populations. And with that, I am going to hand control over to Phil. And while he's getting ready and we're kind of switching over that control, I will let you know that you are welcome to reach out to us with your questions or interest or potential collaborations. Feel free to email us.
Yeah, thanks, Alice, and thanks, Laurie and Solveig, for the introduction. I will zip through the genetics part fairly quickly to try to keep on time a little bit. And I'm going to focus primarily on let's see, where's the forwarding? It's down here. On some of the introductory background information on carrots, colors, and talk rather quickly about some of the genetics of the major color categories. So carrot's an interesting crop because in terms of the written history, almost all that's written is about color. We know that the first thing we would call carrots today as a root crop was first well-defined only 1,100 years ago, which is relatively recent compared to a lot of other crops. And the first colors of carrots were not orange, but rather yellow and purple. And somebody more than 1,100 years ago was brave enough to eat some of these wild carrots in the lower left-hand corner. And we don't know the story of how the white colored wild carrot became yellow or purple first, but we can only guess about that. But for sure, the first colors that were grown were yellow and purple based on written history. And so if we look at where carrots started as a root crop, for sure, there's some possibility they could have been in existence in the Roman Empire, but I won't spend any time on that today. The best verifiable history for the origin of carrots comes from Central Asia, in particular Afghanistan. And from there, carrots spread to the west primarily through Iran and Syria and up to over to Spain by 1100 or so AD. And then started spreading to the east also a little bit later and not quite as quickly. So that these first purple and yellow carrots were recorded up through the 1400s, up and through Europe and Asia. Starting in the late 1400s and into the 1500s, the first orange carrots were recorded in initially southern Italy, southern Spain, and southern Germany. The Dutch are famous for popularizing orange carrots, but probably weren't the first to be growing them. But purple and yellow were still grown in Japan at this time. And white carrots, interestingly, even though wild carrots are white, the first white cultivars showed up only about 500 years ago. So this history is fairly well documented, but we're always open for more information as archaeologists find more information for us. One important thing to bear in mind with the colors of carrots is that the colors give you some good indication of nutritional value, which I won't talk a lot about, but white carrots have a lot of good fiber, but none of the pigments that I'll be talking about primarily in the rest of my talk. The orange color of carrots are most important because these are precursors of vitamin A, and vitamin A is an essential nutrient. Red color is lycopene, the same pigments as in watermelon and tomatoes, and lycopene imparts some nutritional value in some forms of cancer protection. Yellow, some forms of, helps to contribute to eye health. So all of those colors are carotenoids I'll talk about first, but then the other color category is in the middle here with purple along with orange, but the purple part are anthocyanins, which are great antioxidants. So not to talk more about that, but to move on to carotenoids and talk to you quickly about the genetics. So the biochemical pathway of carotenoids are well known, and it was thought for a long time that the colors of carrots were controlled by genes in this pathway. Turns out that's not true. There are genes outside the pathway that control the colors of carrots, but we can see in the pathway that red is the first color, lycopene, alpha and beta carotene showing up in the next steps to give orange carrots, and lutein further down the line. This is carotenes. We'll talk about anthocyanins a little bit later. The experiment, and so initially there were two major genes that were thought to be controlling the difference between white, yellow, and orange carrots. And these are referred to as genes Y and Y2 that were studied quite a while. And I won't go into the details of putting genes on chromosomes. We had a good introduction from what Laurie and Solveig had to say, but we know where these genes are on the carrot genome. So the colors that are controlled by these two genes, Y and Y2, if we start in the upper left-hand corner, this actually looks like it's pale yellow, but it's a white carrot. And these are two dominant genes for these two dominant alleles for these two genes. And so, like as Laurie said, if you have one allele for the gene, it's going to be dominant. And so whether it's homozygous dominant or heterozygous, that for both of those genes, the carrot is white. An unusual color that doesn't show up historically is pale orange. That's in the upper right-hand corner. And in that case, the Y2 gene is homozygous recessive. Yellow is in the lower left-hand corner of this square. And the yellow carrots that first showed up in written history are homozygous dominant for the Y2 gene and homozygous recessive for Y. And orange is one gene difference, one allele different from yellow, in that it's homozygous recessive for both of those genes. Don't need to remember that. It's not critically important, and it turns out that the genes are on different chromosomes, so it makes it easier for us to breed than if they're closely next to each other on chromosomes. But from the standpoint of moving forward to understand the genetics, this was an important part of what we do. So to get a handle on the genetics of the traits, I'm not going to go into details, but to say that in order to map the genes for Y and Y2, we created populations that, contrary to the breeding you heard about from Laurie and Solveig, we self-pollinated plants as Mendel did, because that's the best way to get good Mendelian genetics information. And when you self-pollinate these carrots, you get multiple colors from the progeny. And what these lines indicate is that the yellow carrots in this yellow-white population weren't all the same color of yellow, but there's some variation. The white carrots had a little bit of pigment, but not much. In the other population that was segregating for the Y2 gene, we had dark orange carrots and in pale orange carrots. In that way, we map the genes. And unless there's questions coming up, I'll leave it at that, just to say that that's how we map those genes and put them on the chromosomes. This shows a population that's segregating for the Y2 population. So what happens when we self-carrots and then start genotyping and phenotyping them? we genotype them in addition. Well, we assign the genotype based on molecular markers, gene markers, DNA markers, which again, I won't go into detail about, but it's basically something like the human genome evaluation with 23andMe on a large scale for carrots. So more recently, we discovered yet another gene controlling orange pigments, and I'm not going to go into the details other than to say this came from an evaluation of about 700 carrots of various shades of orange, yellow, and white color, and discovered a new gene on the genome that gives some variation depicted in this slide. It turns out this OR gene is one that is homozygous recessive in most carrots. And so that's why we haven't recognized these other versions of it until this large term study. So then this OR gene is interesting because there's also a gene in cauliflower and melons that gives color to orange cauliflowers and orange melons. So it's a gene that has some family members in other plants. So that gives a quick background on the difference between white, which is doubly dominant for Y and Y2, yellow, which is recessive for the Y gene and dominant for the Y2 gene, and orange, which is recessive for both Y and Y2, as I show here. We could say that orange carrots, we could add the OR nomenclature as well, but it turns out that, as I said before, almost all carrots are homozygous recessive for OR, so I won't be talking about it as much in the next couple of slides. The other color category that is controlled by this collection of pigments called carotenoids, and I should go back and point out that carotenoids are essential for plants, not so much in addition to giving carrots and fruits their color, but also a critical part of the photosynthetic mechanism in leaves. But the other carotenoid I didn't talk about, in addition to alpha and beta carotene in orange carrots and lutein in yellow carrots, is lycopene in red carrots. Lycopene is the most multiply gene recessive trait in that it's homozygous recessive for Y, for Y2 like orange, but then in addition, homozygous recessive for two more genes elsewhere in the chromosomes. So in terms of molecular and genetic information, red is the most recessive of all the colors. That has some implications from a breeding standpoint in that it makes it relatively easy to identify outcrosses to populations when you have a red carrot that may have had some pollen coming in from another source of carrot color, because all of those other outcrosses will give you orange, yellow, or white carrots rather than red. So that's a quick overview of the major genes for carrots, and this gives you the range of carotenoid-based colors. These fat-soluble carotenoid pigments, white being homozygous dominant, two genes, pale orange and yellow homozygous for one of those two genes. Orange for homozygous recessive for the both genes, and then with the OR gene thrown in, we don't note it here. Then red homozygous recessive for three genes. So this is all based on Mendelian genetic evaluation to get this kind of information. It's important to know this because then when we look at the question, okay, well, we have these two major genes that control orange color, but we see a wide range of color beyond orange. These are all orange carrots. The ones on the left probably have about, you don't need to remember the numbers, but somewhere about 50 parts per million carotenoids. The ones on the right up to 400. They're all orange. They're all homozygous recessive for Y and Y2, but there's other genes controlling the pigments. So this would go back to the complex inheritance we heard about earlier. And that's what gives us the ability to breed all the way up to 400, maybe up to 1,000 parts per million. So carrots are very nutritious in that way. Briefly talking about the anthocyanin pigments, we move on from these fat-soluble pigments to water-soluble pigments that gives us great range of interesting color in purple carrots. And we did similar genetic analysis. This shows the P1 gene segregating as well as Y2 in this population. There are actually three major genes that control a purple pigment of importance. P1 and P2 both give a similar phenotype, this fairly dark purple color. P1 sometimes more solid purple than P1, than P2 is. But then in addition to these two major genes that give you purple color for different purple carrots, there's a gene that controls the type of anthocyanin. So again, any one of these genes for P1 or P2, or in fact P3, there's 3 of them, I mentioned 2. If a dominant gene at any of those loci in the chromosomes will give you purple color. And it will give you purple color on a backdrop of whatever color, carotenoid color there is. So here we see a purple color on an orange carrot. Here we see purple color on the 2170 on a yellow carrot, and upper left, a solid purple carrot. So we can't tell what carotenoids might be there because the anthocyanins, these purple pigments, will be masking it. And again, a simple genetic control for that trait relative to the carotenoids. So, but the anthocyanins are not just one compound, but a mixture of 5 different compounds. And they're an interesting group because of their nutritional value. They're all based on a structural background called cyanidin. And you can think of these as sort of ornaments on a Christmas tree in this lower right-hand portion of the molecule, where some of the, some versions of this chemical, this anthocyanin, are acylated, 2 versions of it, some are non-acylated. That's important from a human nutritional standpoint. It turns out that the non-acylated anthocyanins in carrot are anthocyanins that are nutritionally important because they're readily taken up when we eat purple carrots. Those other 3 anthocyanins that are all mixed, all 5 of these are mixed together in any purple carrot, but there's a gene for the balance of acylated to non-acylated, and that controls whether most of them are acylated, whether most are non-acylated. If most are acylated, excuse me, like the bottom 3 molecules, they're less nutritionally important. So something to think about from a nutritional standpoint. They're less nutritionally important because they're not readily absorbed when we eat those purple carrots that are high in these 3 compounds on the bottom of the list. And this is based on some human nutritional studies where people were eating purple carrots. Their blood serum was evaluated for the presence of the pigments. This bottom blue line is a non-absorbed anthocyanin. The green and yellow are less absorbed, and the black and red are readily absorbed. So the purple pigments, again, are important antioxidants, and different purple carrots have different nutritional value, but all have some positive nutritional value. And as I said before, when you look at the genetic control of the color, it's this purple gene will overshadow whatever anthocyanin genes, what other carotenoid genes are controlling the color of the carrot. So here's a purple carrot on a white background, on a yellow background for several examples, and on an orange background on the bottom. So that's a brief overview of carrot pigments. When you're breeding for purple, then you are bringing in a new version of alleles that overlays the anthocyanin colors on the carotenoid pigments. So with that, that concludes where I'm at on my part of this presentation. Should I move right on to the next part of the talk that we're presenting?
Yeah, that would be great. And I just wanted to acknowledge, thank you for putting questions in the chat. We're trying to answer you as we can. And also there's some questions in the Q&A that we'll get to here after Phil finishes this next part of the presentation, and then we'll watch a little video of us actually doing some of the selection and talking through it. And then we'll get to just open up and have some open Q&A time. So go ahead, Phil.
Okay, thank you. So talking more about the incorporation or the movement of genes into traits, Laurie's going to have some comments as we go along and some toward the end. So just talking about the whole breeding process, and Laurie and Solveig gave some good background on this already. We have this wide range of diversity for traits in carrots, everything from wild carrots to this whole very wide array of colors and shapes. The question is, how do we make progress in a breeding program when we're looking at different traits, different genes in a range of carrots? I showed you this slide before with some different words on it, and I gave you some background information on the colors and the genes that control them, and I'll mention them briefly as we go along, but mainly to point out that in addition to these major genes for colors, most of the breeding work that we do, say in an orange carrot breeding program, is for the minor genes that control the amount of colors of the orange pigments rather than than whether it's yellow, white, red, or orange. And so those minor genes are very important. And as was mentioned, it's important then because of that to grow the carrots that you're interested in, if you have the opportunity to grow them in multiple locations, or if you're breeding for your location, keep doing what you're doing, and you'll be able to make some progress over the years by having a consistent selection regime. A couple of traits that are relatively difficult to breed out of carrots are yellow cores, and this isn't a bright yellow core in the lower right, but it's fairly pale yellow core. So yellow cores and green shoulders, if you have them in your breeding population and you want to get rid of them, they tend to be pretty tough to get rid of. The genetic control is not very strong, meaning that there's a strong non-genetic control to it, and it's very difficult to select out of it. Most modern carrots have been selected very aggressively for non-yellow cores, and for non-green shoulders, it's the same story as yellow cores. If you have it in your starting population, probably the best way to get rid of it would be to cross that population with another carrot that does not have green shoulders or yellow cores, but that means starting all over. So these are tough traits to get rid of. If you are able to, good luck, but compared to traits even like the shape of the crown shown above here on the left, or the shape of the tip, which are more easily bred for, green shoulders and yellow cores are tough ones to breed away from. We talked briefly about flavor. As was mentioned for diseases, if you're growing carrots in a place where you get nothing but bad flavor because of the climate, then that may be the best place to breed for flavor, because you're going to find very few good flavored carrots, and you can be assured that when you're breeding for flavor in those growing conditions, you're going to get better flavored carrots than if you're growing in Southern California, as was mentioned, where most everything tastes pretty good. So again, it depends on where you grow the carrots to make progress. Already it was mentioned that if you're going to breed for disease resistance, you have to have exposure to the disease. And here's some alternary leaf blight, cavity spot, and nematodes shown in this picture. We spent a lot of time breeding for nematode resistance. We started, this is a project with Phil Roberts, breeding for nematodes that occur in the soils of California, which is important because it's a big carrot production state, where all of the cultivars we started looking at looked like the ones in the upper left-hand corner that were completely damaged by nematodes. We ran into a cultivar Brasilia from Brazil that had a few roots that were resistant to the nematodes. And so what we did, of course, was to intercross those without the nematode attack, and eventually bred those genes into carrots for the so-called cello or whole carrot market for the large-scale growers, and the long skinny carrots that are used for baby carrots, so that today we now have some level of nematode resistance in these carrots with this breeding program that started actually in the 1990s. So it's not a quick process, but that's an example of how we move forward with breeding for a tough trait. Now we're looking at traits like heat, drought, and salinity tolerance, which are looking to be very important as we move forward in growing carrots, both in the US and around the world. And there's a wide range of variation shown here in fields in Bangladesh and in Pakistan for heat tolerance, drought tolerance, and salinity tolerance. And then finally, and I think Laurie will weigh in on this, breeding for male sterility, which is critical for breeding hybrid carrots. The main point I'll make on this before I show a couple more slides and hand it off to Laurie is that cytoplasmic male sterility means that the genes are in the cytoplasm of the cells of the carrots. And we'll mainly talk about the petaloid type and the brown anther. I'll show you those in a minute. But before we do that, most of the genes, all of the genes I've talked about thus far are in the nucleus, but there are genes in plants, in the mitochondria and the chloroplast, and it's the cytoplasmic male sterility genes that are in the mitochondria. And so those genes are only passed through the mother parent. And what was discovered in the 1950s is carrots that, instead of your normal carrot flowers, like in the upper left here, which have 5 petals and 5 anthers, Henry Munger discovered a carrot with not 5 petals, but 10 petals and no anthers. The anthers replaced by petals. This is called petaloid sterility. There's also another form of sterility where you have the anthers and the petals, but the anthers have no viable pollen. So either way, these are carrots where all the flowers on the plant have no pollen, so they're male sterile. And that's the basis for making hybrids. This is what some actual photographs of petaloid carrots look like. Again, where the 10 petals instead of 5 petals and 5 anthers. And the process of getting this sterile cytoplasm from wild carrots into orange carrots took about 15 to 20 years, so that today we have the starting materials for making hybrid carrots, and they contribute significantly to new carrots being grown for large-scale production. So it takes a long time to incorporate these traits, requires lots of record keeping, but it's now being used for large-scale production on a wide-scale basis. Laurie, I'll turn it over to you for any comments you may have. If you'd like me to go back on slides, let me know.
Yeah, I just a few things to add. I think we'll do a future webinar that goes more deeply into hybrid production and some different ways to do it and what male sterility means. I just want to make sure that folks understand that in carrots, male sterility is a naturally occurring phenomenon, and it is regenerative. You can breed fertility back into it. There are the existence of what are called restorer genes, and this is a whole thing that we're not going to get into right now because we have more to keep going with. But just to know, if you're working with hybrid carrots, or if you want to breed out of a hybrid carrot, you are 99.9% likely to run into male sterility. And it's probably not going to work super well. But also, if you are breeding and you notice that this brown anther or petaloid sterility shows up, don't panic. It's natural. It's not going to drift over into your other plants. You can just either pull that plant out or save seed from it, and know that the seed from that plant will also be male sterile, because as Phil said, that passes through the maternal pathway. Again, we'll get more into that in another webinar, or you're welcome to reach out and follow up with us. But we have this really fun video that is about 19 minutes long and shows us actually doing some selection in the greenhouse yesterday. So I just want to show that to you all, and then we'll open up for Q&A. Go. Hey everybody, I'm Laurie McKenzie from Organic Seed Alliance. This is Micaela Colley. We are here today to talk to you about carrots and carrot breeding and carrot selection. We've got 2 different populations to look at and work with and show you today. This is a very early generation. This is a variety, well, it's a population, but we've given it the name Fantasia. It came from a school project of rainbow carrots that were grown out at our local elementary school with Micaela's children and some other kids who did some color and taste selection on it. So you can see it's really diverse. There's a lot of color diversity, there's some shape diversity, there's some length diversity. So what we're doing right now, this is about the 3rd generation, and we want to keep it really diverse. What we want out of this is actually a rainbow population. So variety that you can plant and you'll get all these different colors of roots. What we don't know yet is how we do that exactly. And so we don't know, some of these colors are more dominant than others. As we mentioned earlier, the purple and the yellow are more dominant. The orange and the red are less dominant. So when we select the roots that we're going to put in our cage and make seed, we're actually going to select less of those than we're going to get when we plant the seed and grow the actual variety or population. So the first thing I'm going to do right now is look at tip shape and tip fill. And what I want is something like this, where I've got a nice cylindrical root. It's blunt on the end. I've got lots of tip fill here, versus this is not what I want. This is a rat tail. It kind of just keeps going. It doesn't really fill in. It does have some nice shape and girth to the root, but I'm going to take that one out because I don't like that. And that is something that we can make progress on just by doing this visual selection.
So Micaela's going to come over and help me, and we're just going to go through and we're going to take out some of those. And as we go, we'll tell you other things we're looking at. This yellow has green shoulders. That's a trait that Phil mentioned that is difficult to get out. So we're going to take that out. It's not as much of a problem, I think, when you have the purples, because it kind of hides.
Some of these, Laurie, I notice have a little bit of rat tail, but you can still see it's trying to fill out toward the end here. And so with a little more maturity, it may fill out. That's different than this one here that just keeps going and tapering and isn't even trying to fill out. So prioritize taking out those roots that continue to taper with no fill.
Yeah, and we know that we've got a lot of roots. We still have more roots in the bag to go through. And so we don't need to worry too much about our population numbers at this point. I like this one. I like the colors. It's nice and thick, but it kind of looks like it just wants to keep going, like Micaela was saying. So let's say these are looking pretty good. Take out a few more. This one looks like it wants to be really long, which is not really what we're going for. And again, this is still really early. So we're maybe going to leave in a little more diversity than we would with an advanced population, but that looks pretty good. So the next step, should mention the other things we're looking at here are not wanting multiple tops. Like you can see, this has multiple tops starting to grow. That is something we want to select away from. That root is so beautiful. It's got a lot of characteristics I like. I might keep it in. We don't like I don't like really thick shoulders. That one's probably not going to grow any tops for seed production. So I'm going to take that one out. All right, so now we're going to go through and we're going to cut them open. It's nice to cut them on a diagonal because that gives you a really nice look at the internal color and the internal characteristics. I'm cutting them down fairly low so that I've got plenty of root left to look at, to work with, to grow from. And you'll see taste. We taste select all of the roots in our breeding work. And I'm actually just going to get rid of those because they just are visually distracting. And I'm actually just going to taste from the roots. So what I'm hoping for with this population is that there's just a lot of really cool and interesting diversity. That there's color variation on the outside, there's color variation on the inside. And we're looking ultimately for this to be a variety where the shape of the carrot is fairly uniform, but the colors are really diverse.
Laurie, I think I'd also select against some of those lateral side roots that are coming out of a few of them. Doesn't look too bad overall, but this one in particular stands out that way.
Yeah, absolutely. And one of the things we mentioned earlier in the webinar is that when you see variation in a trait, when you see diversity for a trait, like Micaela said, these are quite hairy. This one as well. And we also have roots that are not. So we have roots like this that are really smooth, really nice. We don't see those hairs or they're pretty minimal. And we got some that are kind of in the middle. So when you see diversity of a trait, that tells you that there's differences. And that if you select towards one way or the other, you're probably going to be able to make progress. That's a good indication that there's some different genetic combinations happening there. So that's a great point. We'll take those out. And then we would do this to all the roots. And then we would go through and we'd look and see if there was anything in the internal characteristic that we really don't like. Because this is a population where we want a lot of diversity, I don't see anything here that's strikingly terrible. I don't love this white core, but let's leave it in at least for now and see if it tastes good.
So Laurie, you already mentioned this is a diversity mix and your goal is to maintain that diversity, but out of a wide cross like this, you could also choose to take it a few different directions. You could decide, you want to do just the purple yellow and pull those out and keep them separate, or some of the other combinations.
Yeah, absolutely. And that's one of the breeding goals of this project is to actually create breeding populations like this that are really diverse, that we can share out with other breeders, seed companies, carrot enthusiasts. So you could grow this and you can say, wow, I really love these. I really love that color combination. You take everything that looks like that, plant those together, and you start on a path to make a new variety. So yeah, there's a lot of really fun potential here. And because it's still early on in the breeding process and the generation, there's just a lot of different directions it could go. So with that, we're going to move over to the opposite end of the breeding spectrum and look at some advanced maturity.
This is a red variety that we've been working on for several generations. Initially, Dr. Simon Phil created it, crossing red material from India, selecting against bolt sensitivity, which a lot of the red varieties tend to be very bolt sensitive, and crossing it with some orange material, orange varieties that have good flavor and a little less bolt sensitivity. So out of that early cross, which I'm not sure how many generations ago that was, for at least the last 6-8 years or so, we've been selecting toward a more uniform red nonce type variety. These are all selected against bolting in the field. And then at this phase, when we're selecting for the root appearance, again, as Laurie said, in this variety, we are going for that nice round tip fill. You can see some very nice filled out kind of cigar shaped individuals here. So just like Laurie said, we would start out first off selecting out anything that goes the other direction. Boy, there's one that really comes to a blunt tip.
And this one actually is a bit deceiving. I think it would actually be blunt, and it's a good one to select from. But I also want to note that this is more of a physical injury blunting. So maybe while it was growing, it ran into a rock or a hard clump in the soil and started to grow around it. And that's what sort of forced this tip to fill out. I do just have a sense from the rest of the carrot and kind of how it's so wide that this would be well blunted if that tip had filled out naturally. But I think that's actually an injury or an obstruction that caused it to be this blunt.
So I'm just going to take a few out for a little less blunting, even though those are not too bad for a red carrot anyway.
And this one's a little long. I mean, it's nice, but just compared to everything else out here, it's a little on the, this one as well, a little far out on the edge of the bell curve.
You can also see, if you look at the, as Laurie was saying on the bell curve, you can kind of take a look at the whole population and think about, there's some color variability that I can see visually with some of them. This one looks like it almost has a little bit of orange in it. I'm curious what the interior color is. This one here is very dark red. That is a nice dark penetration of red. Some red carrots tend to get this sort of lighter color on the surface. Not bad, but a little less desirable, especially as we're trying to make this a more uniform population. You can really see the difference with those two next to each other. This is the red we're going for. So I would also look at that, as Laurie said, when you have diversity in a spectrum, it also means that you have a lot of room for improvement by selecting against what you don't want and selecting toward what you do want.
Not quite. And this is, we mentioned earlier in the webinar, the different selection techniques, and we have used mass selection on this for the entirety of our breeding work so far. We haven't done any family selection on it because we really haven't needed to.
Well, there's a lot of diversity still in the variety to select for. We have also created separate population from this. We have started creating a new mix of red varieties crossed with each other in order to get, again, to infuse more diversity into a population with the opportunity to then select a little harder for flavor or bolt resistance or any of those other traits. So yeah,
I'll hand you some.
Sure.
This one's a little light.
So with the reds, you also tend to, what we want is a nice deep penetration of color. You can see this white in the center here is a little less desirable. So we tend to select against that. You can compare that. Here's some that show you just a little bit more of a spectrum of that. So there's a combination of things I'm looking at here. I really like this dark red color of this individual root, except for the white center. I like the depth of the red other than that white. And then this one, you see, you don't have so much of that white core. So there's a nice center core. Here's one also. That's a nice one. It's got a nice
A little more white.
my hands are getting full. So we'll select against some. So in this case, I'm going to take this one out because of that yellowish white core in the center. And that one out. And that one out. And we're going to keep these in. There is still some diversity here, some spectrum, but we're moving the population toward that nice interior color.
And I think this is another good example, like that's a little more white in the core than is desirable, but it also has a beautiful external color.
And smooth.
And it's smooth and the shape is lovely. The shoulder is good. The top is good.
Selection is always a balance. You know, you as a mentor of mine once said, breeding is like juggling balls in the air. If you've got 6 traits that you're trying to go for, you can only sort of keep so many things in the realm of what you're selecting against until you get so picky that you don't have anything left. So what are those 6 balls you're going to put in the air? In the case of this one, bolt resistance is key. We want good flavor. We want to have a nice red color. That tip fill is important, and smoothness. So there's my 5 or 6 breeding balls that I'm juggling up in there in the selection process.
And we have done taste selections on all the roots through the entire cycle. We don't wait until we're at an advanced stage to do tasting. As we didn't really get to, but we mentioned with the Fantasia rainbow population, we've tasted those every year. And if you don't have good flavor to start with, it's not going to suddenly appear. So it's something you always want to be keeping in those balls that you're juggling, and it will change and it will respond to the environment and the kind of growing year that you had. And so you always have to keep that little bit of flexibility in there for like, you're gonna find a couple perfect carrots that have everything you want, but not every carrot is gonna be the perfect carrot, and that's fine.
The reds are an interesting one when it comes to flavor too, because there is a lot of diversity in the texture. Some of them tend to be very dense and a little dry, and others are very succulent and crisp. There's definitely differences in the sweetness and the flavor. And what we're going toward with the reds, we work with a woman named Jaspreet who's from India, and she grew up eating red carrots. She never saw an orange carrot until she was 15 years old or so. And so we always lean on Jas's palate for tasting the red carrots, and she describes the red carrots in India being crunchy like an apple. I just heard her crunch right when I said that. So we definitely are leaning on the flavor of the red carrots, wanting that crispy, succulent, and mildly sweet flavor.
Yeah.
And as Laurie said, we taste everything. So literally every one of these roots we will taste and decide whether they stay in the population or if we eliminate them from the population.
Oh, that one has really nice color. The other thing that could be challenged with the reds and the purples as well, is that they tend to have a lot of sort of turpentine and piney flavors, and those are really important. They actually are essential components of kind of what that carrot flavor is. If you have too much, then they start to taste soapy, like tree sap. And so there's always a balance there too. We're looking for texture. We're looking for a little bit of that piney, turpentine, floral flavor. We're looking for sweetness. We're looking for sugar. And kind of, really trying to sort of stay in the middle of the bell curve. And not being, extreme sweet is great, but a carrot that just tastes sweet also kind of falls flat. So a little bit of everything.
That one does look pretty. It's a little more mild. I'll taste it. I haven't tasted any bad yet. So if I did, I'd put them in the other pile.
And like this one, I agree. It's mild. It's a little bit piney. The texture's good. It's not super sweet, but it's good. I would keep it. I like that one too.
So these roots, now that we've narrowed down our selection, these are going to be eaten for dinner tonight. And the rest of them, this is what we would save to plant. And if you've never grown carrot seed before, what we do is, well, these actually were harvested recently. And so they'll stay in the cooler for about 45 days in order to acquire enough chilling or vernalization in order to induce flowering. So we leave them in the refrigerator just like this. 45 days later, we take them out, plant them out in the field, and I'm just gonna bury the carrot with soil into the soil with about that much of the top showing. You wanna leave this little bit of green because that's necessary for photosynthesis and for that apical growing meristem, the growing point of the leaves to then produce a stalk. So the stalk will come right out of the center there and go to flower and make your seed.
And one thing I will point out, and then we'll wrap up, is that these are nice, big, pretty much full-size carrots. You can still do this with carrots if they're not fully mature. They will vernalize. You can still do some flavor tasting and some color tasting. The color and flavor profiles don't completely develop when they're still young, but you can do it. You can do it with carrots that are about this small. Anything that's about the size of your finger or a pencil or a Sharpie, anything bigger than that is generally the cutoff of what you can use to move forward with breeding and selection. So like these are small, but if they were amazing, and this one's pretty amazing, if it tastes good and it meets my other criteria, I would keep this as a duckling and do it for seed production as well. So hopefully that was fun, interesting, inspiring, and we're excited to keep in touch, and we'll keep you posted on how our breeding work goes. Thanks, all.
Can I talk about a couple of questions that came up in the chat?
Definitely.
One that came up is there any association between flavor and color? And there is, in a sense that, as you already mentioned, when I started breeding carrots, orange was the only color and a little bit of yellow and white, and there were no purple carrots around. The purples came from parts of the world where carrots are generally cooked quite a bit or processed, and so nobody was selecting for flavor. Same thing with red carrots from Asia. They usually have historically been cooked, and so the raw carrot flavor that we're familiar with hasn't been selected for. So in order to improve the flavor of those, at that time, brand new colors for the US, we crossed them to orange carrots and then started the breeding process. So it's been a fairly recent effort to improve flavor on purple and reds and a little bit on yellows and whites. And so those colors by themselves, the genes for the colors don't have anything to do with controlling the flavor. It's just that the genetic background that we're moving those color genes into has been a fairly new movement into the better tasting orange carrots in relatively recent history. So again, the association between color and flavor. The other quick question one question I'll answer quickly. We get one cycle of reproduction per year with carrots. And so with these carrots you were growing and selecting, you're vernalizing them, you'll be getting seed by the end of summer. There's no dormancy on carrot seed. You plant it in the fall back in Southern California, and then get the carrot roots next March. So a couple of background comments on some of the questions that showed up in the chat.
And I'll add a little bit of background too as well. We do field trials here every year of all this material as well. And because the growing conditions here, specifically in the Pacific Northwest, are quite different than Southern California. So we go down to the winter nursery, we find some new stuff that's exciting every year. We put that in field trials, and then we look at, is that something that's even realistic to do breeding work on? Because if it's just so minimally productive here, it's not really worth our effort in moving it forward. So we're kind of on an alternate cycle that we produce seed, as we're simultaneously doing trials in the summer here, but then we can't actually trial that seed until the next summer. So our breeding work is always kind of a cycle ahead of our trial work, but we do them at the same time. And I also noticed a question in the chat about were our red carrots that smooth to begin with. The short answer is no. that's my experience. Phil, you chime in if
I'm sorry, what was the question? Repeat it,
were the
Laurie?
red carrots always that smooth?
No,
What
they
the
were
ones
not,
that we
no.
showed in the video were 6220. So a
Yeah,
very
yeah.
advanced population.
Yeah, some of the reds are pretty rough. And I guess going back to the breeding question, if you're growing carrots in northern US, you will, and you don't have a southern location in Southern California to grow your crop, your root crop, like what we were just talking about, you're stuck growing, storing your crop from harvest in September, October, and November up until the next spring. And so in that case, you are truly handling the carrots as a biennial crop. So you're getting the roots produced one year, the seed produced the next year, the roots produced the next year, and then the seed. And so that's how carrots came to be biennials when the Europeans adapted the warm climate carrots for northern growing conditions. But we do handle them on an annual basis because we grow the root crop in the winter time in Southern California and the seed in the summer, or we grow the root crop in Wisconsin in the field, but then the seed crop in the greenhouse. So.
And
Clarification, click and clack here. I would say we do actually do biennial breeding as well. And we have been selecting from the trials that we've collaborated with growers in Vermont, Virginia, Wisconsin, Indiana, Washington. We've been selecting roots and then having from the trials. That's the beautiful thing about carrots. You can do a trial and you can select roots and save them for seed without anything crossing. It's one of the advantages of biennials. So what we've been doing is some of those collaborators are shipping their roots to OSA, and then we vernalize them and plant them out for seed the following year. And then you're able to achieve selection under that environment. So there's screening for material or entries, varieties, and then there's also an opportunity to select. So little combination of both.
yeah, and I'll I've got the Q&A and the chat open. So Alice, you can help if we miss anything, but just a few quick hits on things. One was a question about half sib selection and how do we deal with different seed quantities and qualities. A little bit of self-selection. You know, if one family is a terrible seed producer or doesn't really continue because there's no seed, we don't worry about it too much. It can skew the population if one is a really strong seed producer and one is not. We haven't gotten into that yet, but I would say it's not a huge priority. Is anyone doing breeding for wireworm resistance? Not that I know of. Quick answer, Phil, yes, no?
I'm not familiar with anybody doing any work on that, no.
Does cutting carrots increase the prevalence of rotting upon replanting? Sort of. Depends on how you store them. We aim to store ours with cedar shavings, the ones that Micaela was mentioning that we store over the winter or we get from collaborators. The key to that is doing tasting before you store them in cedar shavings, where everything tastes like pine and cedar. We learned that the hard way. So yes, and in anything that you're storing that long, you should plan to lose some. So we also try to select more in the fall than we aim to actually be able to replant.
If you're listening in from a very warm climate, cutting the carrots before you put them in the ground, as we were showing, can lead to more rot. But that's an unusual situation for most of the people, I'm guessing, on this. But I know my colleagues in Pakistan, India, and Bangladesh often warn against planting cut carrots because they tend to be more prone to rot, but not so much a problem for most of what I'm guessing the audience is.
Yeah.
So can you address a question that a couple of people that have come up in the chat about growing carrots in warm climates like Hawaii? And I know you work with some collaborators, like you said, in Bangladesh, where they don't have winter chilling. And there's a couple of different strategies, I think, of
Right.
either growing in the off season or the more mild season and then vernalizing, or going to an annual crop. Could you mention that a little bit?
Yeah, there's no written history, but I'm quite certain that carrots started out as an annual crop because they originated in Central Asia, which always has had a fairly warm climate. And I'm quite certain that most of the carrots that produce seed in that climate were probably planted. It's about the same time as our Southern California planting. Were seed planted in October or thereabouts, and in the crop grown in the field. But these were carrots early on that were not selected for early flowering. That's one trait we didn't talk about much, but those of us with European ancestry, our European ancestors, because of their cold climates, they were selecting against early flowering because they didn't. If you're growing carrots in Belgium and it gets to be October, you don't want a flowering carrot because it's never going to go to seed. However, if you're growing carrots in Afghanistan in the wintertime and they're going to seed, well, you just let the carrots that are going to seed in the corner of that part of the field go to seed and harvest the rest of them and eat them. And so you get your seed crop and your root crop probably from the same field. You know, these are not mechanically planted and harvested. People go in and harvest what's ready to eat and go with it. And so anyway, initially the carrots, I'm quite sure, were annuals. And a lot of the warm climate carrots grown today, in fact, are annuals when they're grown in our cooler conditions of most of the US and most of the temperate climates. So it's been an interesting history. Does that answer the question, Micaela?
I'll add a slight bit of technicalness to that for here in the Pacific Northwest and also where Phil is in Wisconsin. I imagine we get nights below 50 degrees in the summer. We can have 80-degree days and 50-degree nights, and vernalization accumulation in the plant starts at below 50 degrees. So we can get vernalization, especially in the spring or the late summer during just the nighttime. It doesn't even have to actually be fall. So that's why early bolting does not work well in our climate, why we need to select away from it, because we can get that vernalization requirement met just by nighttime temperatures. And then they start bolting, and then it's not an eating carrot anymore.
Yeah.
We also had a question here about isolation. And you do need a lot of isolation, different distance between carrots to do pure seed production. We didn't mention, but carrots will also cross with wild carrots. Phil mentioned that a couple of times in terms of moving over traits. So if you have Queen Anne's lace, that will cross with carrots, and you'll get woody white roots showing up in the next generation, which are easy to select out, but not something you want to be selling for selling seed. We use pollination cages, which are just big mesh cages, and we use blue bottle flies as pollinators that we buy from a farm called Forked Tree Ranch in Idaho, and they just ship them to us overnight. We've done a little bit of playing with outside distance. We have Queen Anne's lace. Sorry, I keep looking out the window because I'm actually at the farm. We have Queen Anne's lace probably 200 yards from where we do our carrot production. And we did see some crossing this last year when we did a small planting outside, and we had corn and sunflowers in between them. So they had some tall plants in between, but we still did get some crossing. So.
I just want to interrupt for 1 second, just to say that we had several questions in the queue all through the webinar about whether or not this is being recorded. I just wanted to say again that it is being recorded, and the recording will be available on the eOrganic YouTube channel as well as on the eOrganic website at eorganic.org under webinars. And Micaela and Laurie are going to send me some resources, including some information about an upcoming carrot conference. So I don't know if you just want to briefly mention that as well, but we have also a whole bunch of questions. So.
Great. I'll just mention briefly, there will be a follow-up email. This webinar will be shared along with a number of other resources. Maybe if Alice or somebody could put the Carrot CIOA website in the chat. That website, which is on eOrganic about this project, has already a collection of resources that you can follow. The Carrot Conference is in August. It's amazing 3 days of all carrots all the time with carrot geeks from all over the world. we will definitely be there. It's affordable. If you can get yourself there in Mount Vernon, Washington in August, it's a lovely place to be. The last thing I'll mention is that part of this project, the Carrot Improvement for Organic Agriculture project, is a participatory approach to research. And so, as we've mentioned a little bit, we do collaborative trials with farmers, with seed companies. We are developing populations that are interesting and useful to breed from. There's sometimes a limit of our own capacity to coordinate how many people and how many trials are going on, but we are creating a group on the Organic Seed Commons website, which is a platform that OSA hosts where we have events, webinars, classes, discussions, but also just ongoing chat threads. So it's complementary to eOrganic, where eOrganic is a great archive of all of these resources. In the Seed Commons, we will be having more discussion groups and engagement from peer-to-peer of farmers that are working with carrots that have questions they want to ask or results that they want to share with each other. You will get an invitation to that group in the email that comes as a follow-up.
And Canadian growers in Canada, if anybody's out there, I'm coordinating the Canadian Organic Vegetable Improvement Project. We're doing carrot trials, both orange and red this season. So I'll put my email in the chat and please get in touch if you would like to be part of that. Yeah, love to have more carrot action.
All right, I'm going to do some rapid fire Q&A answering because you all have some really excellent questions. And I know we're not going to get to all of them. We're not going to get to all of them in depth, but there are a few I feel like we can answer fairly quickly. What about the storability of red carrots? Is it always worse than orange or white carrots? Not in my experience. My experience has been there's not really.
The Asian reds are tougher. If you got the seed directly from Asia, not the ones that have been bred here, especially from South Asia, they store poorly. But as a group, but not all of them, but most of them do. Let me answer one question very quickly too about seed-borne diseases. Kat asked is two of them, alternary leaf blight and xanthomonas. Xanthomonas can be fixed by heat treating the seed, dipping the seed in hot water, 50 degrees Fahrenheit for 10 minutes. So it's a controllable seed-borne disease. It's a bacterium. The other one is alternary leaf blight, which is not very easily controlled. The only control is growing the crop away from a source of the disease. So,
yeah. If you are breeding for high latitudes, is there a disadvantage for making selections at low latitudes? Yes, sort of. I would say that's why we try to do kind of this collaborative root selection where we're doing some root selection from our own trials here in Washington and combining those with the roots that we're bringing back from El Centro in California, because we realize that's different environments. So we're trying to do some multi-environment selection, also working with collaborators in Vermont and Virginia, Wisconsin, and Indiana on some of that. So
Quick question, follow up on that though, Phil. Carrots are not day length sensitive, are they? Yes.
Not much. There's some evidence, but most of it's temperature, vernalization temperature.
So the latitude is more related to different environments.
Yeah. The problem with the selecting in low latitudes is that the production and flavor tends to be better, and you get spoiled if you're only growing in those conditions. If you want to really get carrots that are going to work in higher latitudes, you have to do most of your selection there because of the diseases and the shorter growing period.
Yeah, and bolting, for sure, for bolting sensitivity.
Yeah.
How do you select for leaf health with mass selection? When we do our initial selections, we dig with tops on. And so we just don't select ones that don't have good leaf health. That's what we do.
Yeah. The other thing is with leaf health, it's usually alternary leaf blight we're talking about, and be sure you're starting your selection before harvest time. Because the diseases start coming in earlier, and you want to see when it's coming in, and at least take notes on if you've got some various cultivars you're testing or your own selections you're testing. You want to take notes on that pretty early because of the timing of the inception of the disease. We get if you got lots of crops in your field, you're looking at your carrots regularly. But those of us that focus on carrots tend to plant them and don't look in the field in the middle of the season as much as we should. So
Another thing to think about with tops is early vigor and canopy cover, because that can affect how much time you're going to need to put into planting. Also, top attachment can affect how easy they are to harvest and whether you can sell them bunched with tops, for which you can sometimes get a higher price.
And that early vigor translates to less weed control necessary. Which is a huge deal for organic growers.
Absolutely. This is always a question. Ray asked, what is the minimum population to avoid inbreeding depression? Always a squiggly answer too. My answer is it depends. It depends on if you are taking roots from a population that already is really diverse and has a lot of genetic diversity. You can probably get away with a smaller number of roots. If you're more likely to run into inbreeding depression when things are not very diverse and you've got a lot of already what's the word I'm looking for? Uniform genetics.
Homozygosity.
And sometimes it works fine. So generally speaking, with things that are strongly cross-pollinated, you want to be more in the 20, 50, 100 individuals range. We've definitely done breeding work with 15, 20. I personally feel more comfortable with smaller numbers if I'm in an earlier generation where I'm confident there's already a lot of diversity in the background.
For those that maintain genetic carrot germplasm and try to maintain all of the diversity, you want to be growing a couple hundred carrots and use a minimum of 50 plants in your seed production, representing the breadth of the diversity of your starting population. Obviously, when you're breeding, you tend to narrow that down, but 50 is a good number. On the other hand, there's any number of carrots where you can have a whole open pollinated cultivar that started with 1 or 2 carrots in earlier generations.
We had an earlier question that got missed from Joseph about fertility in carrots. And I think we can attest firsthand that carrots respond quite well to nice, healthy, fertile soil.
Fertility matters.
And deeply cultivated.
Soil texture as well.
And I would say, in my experience, carrots tend to be more environmentally responsive than a lot of other crops, and that makes tasting even more important. And also, you get to know it over time. I'm now personally about, how many years am I into this? 7 or 8 years into this, and I now feel like I'm hitting my stride. Like, okay, I understand flavor profiles. I can make pretty quick decisions. I understand, okay, I've tasted this variety roots from a couple of different locations. I understand what its breadth is. I kind of can make connections between this was a really ideal growing environment. This was not. I can taste the stronger flavor, but I can associate that with the growing conditions. So part of this is all just like, it's just getting familiar with your material and getting to know it and getting that sense of intuitive understanding from developing that relationship and watching what the carrots are doing, interacting with them, engaging with them, and just really learning from experience. I know we're out of time. Alice, I feel like there's one more question that's really
we can go for another few minutes. I mean, as long as you're not in a rush.
We answered that one. There was one about the commercial carrot industry is the commercial carrot industry moving away towards emphasizing orange carrots? Why has the focus been on orange carrots for so long? My personal passion, if you haven't picked up on it already, is purple carrots. I just get really, really excited about purple carrots, and especially the purple carrot combinations. So I've been especially attuned to the colors, specifically purple. And I've noticed with the industry folks that have been coming to the field days at our winter harvest, there is an increasing interest and an increasing investment in colored carrots.
And as we get better flavor and production quality of the so-called novel colors, purple, yellow, red, white, you'll have a greater diversity and you'll be able to grow the crop more reliably. The big focus, almost all of our carrots until very recently have strictly come from Europe. and that was an orange. Why carrots? But it's a whole nother seminar. Why orange carrots got to be popular in Europe? We don't know exactly. It's a quick answer, but for whatever reason, orange became popular, and that's what the U.S. was built on pretty much completely, except for a little bit of yellow Belgium and white Belgium up until the 1950s. That's just, and actually orange is the most popular worldwide yet today. But as we get more diversity in the breeding populations of different colors, we'll get better varieties in there.
Vernalization requirements 6 to 8 weeks.
For European derived carrots. Yeah, if you've got something with earlier bolting, it's going to be shorter period of time, but yeah.
Interacting with carrots, I just mean tasting them a lot, specifically for me with that PYP population that I spoke about the breeding work with. I had no intention from the beginning of coming up with a lighter colored, more yellow, dark purple core. It just happened. And I feel like the carrots showed that to me. And there's some really amazing flavor there. And I just followed that. And so engaging with the carrots, letting them show me new things and following that one is really interesting.
Yeah, kind of getting an intuitive feel for which traits travel together. And once you have a feel for that, you can tell when you're seeing something new or different that you might want to really hone in on.
How do we tell when a carrot is ready for harvesting? I dig it. I take out shovels and just dig it. And
that's the only way. Yeah. We haven't got ground penetrating radar to take that job over for us yet.
Not yet.
We're working on it.
Let's see.
I see a question on why do you tell yellow from white? Yeah, the yellows of carrots often are not very significantly yellow. It's kind of a pale yellow. Well, if you're breeding hard, you breed for a darker yellow, it helps. But my theory is one theory of why orange carrots became popular is because of this question. It's easier to tell orange from white than it is yellow from white. But that's, like I said, save that for another presentation.
I'm just wondering when that red carrot that you showed in the video will be available. Do you have any plans to release it?
Yep.
Good.
I would say we are increasing seed on it now, and it has been formally released by the USDA. I will also mention the fact that Dr. Simon's program does not patent or put any kind of restrictive intellectual property on any of the releases of seed coming out of his program. And we will have more press and information about that. Generally from his program, you can request small packets once we have enough quantity for distribution and we're prepared for that. So it is already headed out the door.
And if you're working on a participatory trialing of things, all of those carrots are open pollinated. You can start your own breeding program with any of those carrots. They're free for whatever use you want, even if they're not formally released. So have at it and become a carrot breeder.
And in terms of that red carrot, there's a sister line to that one that's very similar, although slightly more pointy and less blunt, that is being currently produced for a commercial seed release next year. Its name is Carnelian, and it will be available through High Mowing Seeds. If all goes well with seed production this year.
Just want to mention again, the organic seed comments. Rebecca put a link in the chat and we'll include more information about that in the follow-up email. And wanted to mention that is a space that we're convening for carrot enthusiasts across the borders. So that is a space that we will be very much continuing to coordinate and engage with the Canadian producers in partnership with Solveig, as well as folks in the US. I often forget that Canada is not our bioregion. Well, it is our bioregion. I forget the borders there.
Yeah, be more convenient if it wasn't.
Yeah. One quick question, Phil. I started typing in the chat and I thought, I'm going to let Phil answer that. Dylan had a question about the alternaria being seed-borne as a disease and the fact that we do alternaria selection and seed production in Wisconsin as part of our program for improving the alternaria resistance, which is part of one of our initiatives. How do you manage the seed-borne factor? Is that not significant enough of an impact that it is prohibitive in your seed program? Because I know you regenerate a lot of seed there.
Yeah, everything we grow has had a xanthomonas wash, and I don't know that that cleans off too much alternaria, but we don't see this. So when we're looking at alternaria in Wisconsin, yes, there is some seed-borne, but there's so much natural alternaria in the Wisconsin growing region that probably our feeling is that the seed-borne portion of it is insignificant. It's mainly coming from the other carrots in the area.
That's hot water treatment, 50 degrees for 10 minutes.
For 10 minutes, yeah. That's for xanthomonas. Yeah, it does not control alternaria. It may wash a little bit of it off.
50 degrees Fahrenheit.
50.
Yeah, 50 Fahrenheit for 10 minutes. Yeah.
Not Celsius.
That would be really, really hot for this.
Okay, interesting.
Also, there's been a couple questions about mesh. The mesh that we buy custom-made pollination cages from Redwood Awning Empire in California. They're fantastic. We just use mesh that excludes flying pollinators. I can't remember what the mesh size is. But again, we use blue bottle flies. So we're trying to exclude larger insects. And they can make them in any size and any orientation. And if you take good care of them, they last for a really long time. And they're not too terribly expensive.
I misspoke. It's 50 centigrade. I'm sorry, I wasn't paying attention. It's 50, yeah.
Fahrenheit would not make sense.
Right, that would be
I'm
cold.
sorry.
That
Yeah.
would be cold.
There wasn't
Yeah.
even logic there.
As it gets written up, I'll make sure it's clear because it's
Cold water bath.
50, yeah, right, yeah. Duh. Sorry.
Like a sauna, a dip.
Well, cheers. I happen to be drinking carrot juice. It brought toast to everybody. I didn't even
I think-
not on purpose.
I think we should start a carrot TV show here. This is so much to say about carrots.
Oh my God.
Yeah. That's what we'll do in the seed com is it'll be carrot coffee hour or carrot juice.
CTV, somebody
Carrot
said.
juice happy hour.
Okay, well, thanks everybody. I think we're out of time, but I'm sure the presenters wouldn't mind if you contacted them. And then also, you can join the Organic Seed Commons and discuss carrots for the indefinite future. And you can learn about the Carrot Conference. So we'll be sending you some more resources within the next 24 hours or so. And we'd also be very grateful if you could fill out the follow-up survey, which you will receive probably in a separate email from that resources email. So thank you all of these presenters. That was really interesting and informative and entertaining. I look forward to future webinars on this project. And thank you all. And thanks to everyone for the great questions and discussion and for joining us today.
Yeah, thank you all for being so engaged. It's really fun to interact with all of you.
Yeah.
Energy.
Okay.
Yeah, thanks very much. Sure appreciate it.
Thank you, Alice. Always a wonderful
Yeah,
hostess.
thanks.
Thank you.